Welcome to FOCUS Immunology. I will talk you through a quick tour about FOCUS Immunology's infrastructure and services. FOCUS Immunology is located in Heidelberg Biotech Park in the campus area of Heidelberg Medical University. It operates a fully equipped tissue culture and cell biology facility with instrumentation for flow cytometry, a multi-format reader for ELISA and fluorescence-based assays, and molecular biological technologies, in particular polymerase chain reaction. This infrastructure is used to provide a full range of immunological assays to the pharmaceutical industry. To summarize a few major applications, FOCUS Immunology performs cytokine release assays for safety assessment of biologicals. Immunogenicity testing is a major area of activity. Functional testing of immune cells for mode of action analysis of immunomodulatory compounds. Immune monitoring of clinical trials, be it single-center trials or even multi-center trials at the international level. And combination of assay systems for pharmacokinetics and pharmacodynamics assessment of biologicals and biosimilars with immunological activity. And FOCUS Immunology laboratories are GLP certified. The next slide provides you with an idea about the location of FOCUS Immunology. Here you see the River Neckar, Heidelberg, the Old Town, and here the Heidelberg Castle. And in the foreground you see the campus area of Heidelberg University Medical Faculty, and the Technology Park Heidelberg is integrated into this campus area. The Heidelberg Technology Park is an international life science park, the center of the Heidelberg Biomedicine Cluster, and the park is closely related to the Heidelberg University and to the world-famous research institutes located there. Here a close-up view on the building in which FOCUS Immunology is located. It's called Im Neuenheimer Feld 515 in the 4th floor. A few words on cytokine release assays for safety assessment of biologicals. These assays are set up as a combination of cell culture, stimulation of cells, and cytokine detection. Either whole blood may be used or defined cellular subpopulations. And the protocols used by FOCUS Immunology and suggested to the sponsors consider the requirements that have been developed after the TGN 1412 incident. As mentioned before, a major area of FOCUS Immunology's activity is immunogenicity testing according to EMA guidelines. The services comprise the full spectrum from assay development to assay validation and finally GLP compliant performance of testing during clinical trials. The EMA guidelines require a sequence of different assay systems, starting from screening assays to confirmatory assays, and also including assays for neutralizing antibodies, whereby such assays require that in many cases biological test systems are used to measure the biological activity of the antibody. FOCUS Immunology has experience in setting up various ELISA formats, like the well-known bridging format, direct binding, and pediative assay formats. A very important point to mention, and which comes more and more into the focus of the regulatory authorities, is cellular immunity induced by biologicals. And for this, FOCUS Immunology has a special experience and special expertise. And so cellular immunity testing can always be discussed with our experts at FOCUS Immunology. Additional information about immunogenicity testing is available at our website. Towards the end of last year, FOCUS Immunology has organized a workshop on current aspects of immunogenicity testing of biologicals, and experts from Merck Darmstadt and the Paul-Ehrlich Institute provided their insight, and summaries of these presentations can be download it at our website. Here you see the starting page of the FOCUS Immunology website and the reports of the presentations from our latest workshop on immunogenicity testing are posted here. Here is the presentation of Dr. Büttel of the Paul-Ehrlich Institute, Dr. Liedert from Merck Darmstadt, and Dr. Bruins of FOCUS Immunology. If you click here, a separate window opens and you find here the summary of the presentation, including figures in which the most important information is summarized. Cell-based functional assays provided by FOCUS Immunology comprise proliferation assays, and we are currently operating 2 non-radioactive assay methods, either a metabolic assay based on the measurement of ATP via luminescence, and 2 fluorescence-based formats, the CFSE assay and brome desoxyuridine based assay. FOCUS Immunology has experience in intracellular cytokine staining, ICS, and in particular for the multiparametric analysis for detection of multiple cytokine producing cells, which seem to play a major role in the cytokine storm incidents discussed recently. Apoptosis assays are available, be it caspase 3 or 7 induction, annexine 5 assay, or the analysis of cell morphology that allows to follow apoptotic events. Cytotoxicity assays are available, 2 non-radioactive formats, LDH release and protease release. NO production can be measured, histamine release, and recently we have established assays for evaluation of innate immunity parameters via stimulation of the so-called Toll-like receptors. We call them TLR stimulation assays. And also for this assay there is a case study available at the FOCUS Immunology website. There you can get more information on this type of assay. For information on innate immunity testing capabilities at FOCUS Immunology, we have posted so far 2 information resources. First, under case studies, innate immunity testing, we describe a typical experiment for testing of innate immunity aspects via stimulation of so-called Toll-like receptors. This is shown here. If you click on the picture, you see an enlarged view, and peripheral blood mononuclear cells from 2 donors were stimulated with ligands specific for the distinct Toll-like receptors, and the stimulation is quantified via detection of TNF-alpha in the supernatant. And you see here quite significant differences in stimulation, for example for TLR4 between the 2 donors, and TLR4, TLR7, TLR9 result in lower TNF-alpha secretion. That's why we have divided the figure with 2 different ordinates. Another resource is a so-called expert report, which you find here. In this report we have summarized specific aspects of innate immunity system function and concepts and case studies with a special emphasis on their use in exploratory clinical trials. You may wish to order and obtain your personal copy by typing in your email address here, and in a double opt-in process to avoid misuse of this process, you will obtain in an automatic manner your personal copy of this report. Lymphocyte stimulation assays provide the basis for most of the assays offered by FOCUS Immunology, and it is important to note that we have a group of defined healthy blood donors which can be recruited for testing under a standing ethics committee vote. Immune cell preparations range from whole blood via peripheral blood mononuclear cells, PBMC, which are isolated by density gradient centrifugation, up to highly purified cell populations, and FOCUS Immunology has established several protocols for purification of these subpopulations via so-called no-touch methods, which means that naive, non-stimulated cell populations can be generated and used for testing. Antigen-specific stimulations of these cell populations are possible, be it by viral peptides like cytomegalovirus peptides, or bacterial products like PBD, which is a tuberculin preparation, tetanus toxoid, or influenza peptides, again viral proteins. Other antigen-specific stimulations can, of course, be established. It is important to note also here that we have in our blood donor population also such donors which have a high immunization status against such antigens, so that reasonable stimulation situation can be used for the testing. Of course, polyclonal stimulations can be done by using, for example, antibodies. Anti-CD3 plus CD28 stimulation is typical method used. Mitogens can be used like phytohemagglutinin or lipopolysaccharide. Low molecular compounds can be used like PMA and ionomycin, or so-called superantigens like bacterial toxins, certain bacterial toxins. And for the functional readout after stimulation, we have different methods available, like proliferation, mentioned already before, cytokine production and release by using ELISA and to measure the cytokines in the supernatant, ELISpot and intracellular cytokine staining to measure and enumerate cytokine production at the single cell level, and even with ICS measure multiple cytokines in individual cells. Induction of apoptosis may be a readout. Cytotoxicity also. So a few pictures to provide you with an idea of the laboratory of FOCUS Immunology. Here you see the multi-format reader for ELISA and fluorescence. Molecular biology equipment and technology is established. There is a possibility for liquid nitrogen storage of cells, cell culture, and flow cytometry. If you are interested in more information or have questions or want to discuss your specific product, please contact either Dr. Eddie Bruins, who is head of the FOCUS Immunology Laboratory in Heidelberg, or Dr. Karl Michaelis, who is head of business development of Nuvisan. Thank you very much for your interest in FOCUS Immunology.